So the next speaker in this lineup is Jared Kress. Jared is an analytical scientist at Merck, where he is responsible for developing, optimizing, and implementing quantitative methodologies using targeted MS metabolomics methods. He is also involved in the development and characterization of biotherapeutic productions, such as with cell culture media, or CCM. Today, he will present the latest work at Merck in the area of routine analysis of CCM components utilizing a targeted LCM-SMS method and stable isotope labeled standards. Jared, the floor is yours.
All right. Thanks, Andrew. Good morning, everyone. I am Jared Kress, and today I am excited to talk to you guys about my work in cell culture media characterization and how we are utilizing Cambridge Isotope Labs MSK QRES kit in our analysis. And it's an isotopically labeled standard, which I will explain in more detail shortly. But before we dive into it, I would like to provide a little bit more background on myself. I've been at Merck now for 5 years, supporting large molecule process characterization, where I've had the opportunity to be a part of a team focusing, like Andrew said, on developing and implementing conventional and novel analytical technologies. In my role, I have a specific focus on mass spectrometry-based workflows or metabolomics workflows, where we kind of provide support for process robustness and understanding of our vaccines and biologics. In the talk today, I will highlight the development of this targeted triple quad mass spec assay, which we are utilizing across multiple platforms here in detecting and quantifying metabolites in cell culture. So for the next 10 to 15 minutes, we'll highlight that work, explain why we entered into this space, the type of instrumentation we are using, and how we are incorporating this isotopically labeled standard into our analysis. Just a brief agenda for topics today. Okay, first, would like to highlight, sorry. So Cambridge Isotope Labs has developed this QRES kit, which stands for the quantification, retention, and system suitability to enable metabolite quantification. The kit contains an isotopically labeled standard mix, which provides many uses. It can be used as a quality control check for your experiments, system suitability to track performance over time. It can also be used for instrument qualification as well. The kit is comprised of a broad range of metabolic compound classes, which we find suitable for analyzing chemically defined media, which is typically used within biomanufacturing processes. During the development phases for our assay, we use this kit to assess matrix effects and extraction efficiencies with this standard. And a big part that this standard plays in our data analysis is what we call data normalization, which we base on functional and structural compound properties, which I will get into later on as well. And some of that work was highlighted in our publication, which you can find in the Journal of Chromatography A, and is also linked here as well. So this is a breakdown of the composition of the mix. You can see, so it does come as 2 vials. The kit contains 2 vials in a lyophilized form. With this kit, you also get documentation on experimental procedures, methods that you can apply in your own lab, as well as those results. The concentrations are listed here. That formulation is the result of a 1 milliliter reconstitution in each individual vial. It's very stable. Again, multiple uses here, and which are outlined with the kit as well. Next, I will discuss some of the key concepts with cell culture media analysis, and why it is of major importance in the biotherapeutic process. So let's first understand that cell culture media characterization refers to the process of analyzing and understanding the properties and components used in the growing of cells. So cell culture media, whether chemically defined or undefined, is providing the essential nutrients, the growth factors, and the optimal environmental conditions necessary for cells to survive, proliferate, and function properly. Characterizing the media is critical to ensuring the cell's health, reproducibility, and overall yield at the end of the process. In addition, spent media analysis, so that's after cells have consumed or produced byproducts, that's also a vital part of upstream process development and optimization. And here are a list of a few other areas where we are employing this analysis, right? So we find it crucial to monitor lot-to-lot variability of vendor-supplied media. So before we bring or introduce components into a process, obviously those components are vetted before introduced. But what can we do to, what's a check for ensuring that there's minimal variation from lot to lot or new material? So that's one area where we're using this analysis as a pre-check. With this, we can also understand cell culture dynamics over time, and as well in spent media. We can determine optimal feed strategy and harvest endpoints. For example, whether your process takes hours or days. Monitoring the output over time. Maybe we can cut down on some of those times to incur cost savings and to speed up the process. And lastly, we can also use this to troubleshoot faulty procedures. We've been involved in many investigations where we've seen abnormal growth kinetics, and one of our analysis is just one tool that the process teams can use. Here, I want to highlight what some of the tools are, being used in the field for cell culture media analysis. For example, there's online monitoring, such as Raman, where the tube or where a probe is inserted into the bioreactor. At-line monitoring tools, such as YSI and NovaFlex, are close to the shop floor bioreactors and can provide quick results, but the coverage is limited compared to other tools. But with offline tools, such as NMR, UPLC methods, including LCMS methods as well, the coverage is expanded. But for these tools we have listed here, we've received feedback suggesting some inconsistencies with the data, as well as reproducibility issues. So given the complex composition of chemically defined media, the need for accurate and efficient detection methods remain unmet in certain situations. So therefore, we have developed a 20-minute reverse phase LC-MS method, which operates in both polarity modes, and we can detect up to 110 metabolites with this method. Here I would like to highlight the advantages of triple quadrupole mass spec in cell culture media analysis. And again, I'll explain more of the instrumentation that we are using. But for those who are familiar with triple quad mass specs, you understand the importance of high sensitivity. So providing improved detection limits, making it ideal for detecting very low levels of compounds. And you can understand why this is crucial in the field of biopharmaceuticals, where there's always a need for, detecting or understanding those low level concentrations of components in the media. Triple quad also provides, superior selectivity. So the tandem, MS uses 2 stages of mass analysis where the first quadrupole is isolating those target ions, and the second one is filtering for your product ions after fragmentation, enhancing that selectivity. It also provides quantitative accuracy. So, precise quantitation is necessary. So the triple quad is widely used for this aspect, providing highly accurate and reproducible measurements. We find, that it also is reducing matrix effects. So it has a better handling of complex matrices, interferences from other components. So it improves the reliability of results, especially in various types of matrix. Very versatile. So it has this multiple reaction monitoring technology. So it allows you to monitor multiple target ions simultaneously, which you can understand is very important. Right? We want to understand how much and what is present in our media. And this is a very, significant tool for us as well. And lastly, lower background noise. So, cleaner spectra, limiting the questionable data, low background noise, etc., leading to clearer spectra. Here I will dive into more of the method information, which can also be found, in our publication. So as I had mentioned, the CCM analysis method was developed on a Sciex Triple Quad plus mass spectrometer, which has many key advantages, which I had explained. So the way this technology works is there's a combination of 3 quadrupoles to improve that selectivity and sensitivity. So your Q1 and Q3 quadrupoles, those regions act as mass filters, where Q2 acts as your collision cell for fragmentation. This setup allows for the MRM multiple reaction monitoring, making it ideal for precise quantitation of analytes. The Sciex 6500 Plus does have a reputation for high sensitivity, detecting analytes in low concentration ranges. And there's many more technology advances, within this platform. I think too many to dive into. Here, I would like to highlight the robust peak separation within the method. So samples are injected onto a Phenomenex Kinetics F5 column. It has a very diverse internal chemistry of the column. It's an excellent choice for polar and nonpolar compounds, as well as other types of methods that include, halogenated, conjugated, or isomeric compounds. Other advantages include, better resolution, higher efficiency, and easy method-to-method transferability. The LCMS method is a 20-minute method with a 15-minute gradient. You can see pretty significant separation. This is just a handful of metabolites here in the chromatogram, but you can see this very specific. Again, that MRM technology is allowing us to pinpoint on a specific region for a specific compound. This slide here highlights the diverse or extensive coverage of media components that we can identify across multiple different types of metabolite classes, such as amino acids, vitamins, nucleic acids, carbohydrates, and a list of other components. So a very diverse panel. The next slide, I'll explain. So how are we quantifying the metabolites in cell culture media? So given the nature of the components, we thought it was necessary to find a standard mix that mimicked the diverse makeup of the media. So as part of our sample prep, we are spiking in this isotopically labeled standard into the sample matrix, and we are monitoring these components listed here in the table, alongside our unknown panel. Post-sample acquisition data analysis involves normalizing our data to provide a relative quantitative value. To do so, we've assigned a certain isotope standard from this list to a subset or a group of analytes that are similar, in retention time as well as molecular structure. So this analyte peak areas to internal standard area ratio is what we call our normalization ratio. So we provide a ratio value as a relative quantitation. But the diversity here allows us to assign, specific isotopically labeled standards to a large list of unknown components. So the next few slides, I will dive into some of the applications. For example, during the development of this assay, we assessed retention time reproducibility as well as peak area reproducibility, right? So using a scheduled MRM method, it's critical that we have reproducible retention times. And for the isotopic standards that were spiked into media, we monitored those peak areas over time, just looking at stability. And for both types of analysis, we found CV values relatively low, which is showing great reproducibility for both. In this case study, we were analyzing lot-to-lot variability of production media that was manufactured at different sites. So we highlighted here that the amino acid data was higher in lab formulated media, which was tied to faster growth kinetics. So we could see that in lab formulation, the cells were behaving and producing byproducts or growth much faster than that of other sites. So here we can just show that the teal color was almost doubled compared to a couple of other sites. That's based on the formulations used and things like that. On this slide, we were interested in comparing different bioreactor tanks within one facility. So samples were collected at harvest time point, which in this case was 40 hours, post inoculation. On the PCA plot on the left, you can see tank number 19, which was a control in this case, showed the largest variation compared to the other tanks in the study. The components in tank 19 were lower than the rest, as shown in the heat map on the right. So tank 19 had extremely less or lower concentration of components, compared to the rest. And those values on the right side are those normalized values that we are generating with the isotopically labeled standard.
1 minute to go, Jared.
Okay. Last slide, final, thankfully. So again, another showcase here. We can monitor, the consumption of amino acids over time. So again, where can we see maybe a drop off or an increase at a certain time point? How is that affecting, overall yield and growth kinetics, post harvest? So some, conclusions and path forward. This cell culture media characterization method, we're able to monitor up to 110 metabolites established on the Sciex 6500 mass spec. We are using the QRES kit, which is an isotopically labeled standard, to provide us a normalization value. Some future work, includes applying this method to build some predictive process models to support process characterization and validation studies. Other areas we're exploring, we want to expand our panel. So we're looking into high-res mass spec for more of an untargeted analysis and using the QRES kit as a standard in that, as well as an instrument check. So that concludes the presentation.
Thank you very much,
you.
Jared. And very enlightening talk. We appreciate you sharing your CCM characterization approach done at Merck. In the interest of keeping this webinar on track, though, we'll move forward to the next speaker, and I'll ask that you address the questions in the Q&A box. There are a couple from your talk, a couple different attendees. But thank you once again.
Thank you.